Even though what we heard just now was all about the bad side of the microbes, but do you know that actually there are certain microbes that can limit the effect of climate change? Microorganisms are very important to use greenhouse gases as energy source and build their cell. Therefore, when microbial communities and biogeochemical cycles are linked together, it is a good mechanism to solve climate change. First, we will talk about microbial communities and carbon cycles. Secondly, microbial communities and methane cycles. And lastly, microbial communities and nitrogen cycle. Firstly, microbial communities and carbon cycle. Microorganisms are part of a larger cycling of carbon that occurs on the global scale. Much of the carbon that enters the carbon cycle is carbon dioxide. This form of carbon exists as a gas in the atmosphere, but before it can be incorporated into living organisms, it must be transformed into usable organic form. The transformative process by which carbon dioxide is taken out from the atmospheric reservoir and fixes into organic substances is called carbon fixation. The well-known example of carbon fixation is photosynthesis, a process by which energy derived from sunlight is harnessed to form organic compounds. Photosynthetic algae are important microorganisms in this regard, and chemoautotrophs are mentioned. Primarily, bacteria and archaea are capable of carbon dioxide conversion into sugar form available for cell building. Some organic carbon is returned to the atmosphere as CO2 form during respiration. The rest of the organic carbon may cycle from organisms to organisms through in food chain. When an organism dies, it is decomposed by bacteria and its carbon is released into the atmosphere or the soil. CO2 dissolves in the water at that time algae, plants, and bacteria convert into organic carbon. In anaerobic environments, microorganisms can cycle the carbon compounds to yield energy in a process known as fermentation. For example, green and purple sulfur bacteria are able to use the energy they gain from the degradation of a compound called hydrogen sulfide to degrade carbon compounds. Other bacteria such as Tuobacillus ferocigens uses the energy gained from the removal of an electron from iron-containing compounds to convert carbons. The anaerobic degradation of carbon is done only by microorganisms. This degradation is a collaborative effort involving numerous bacteria such as Bacteroides succinogens, Clostridium butyroacum, and Centrophormonas species. These bacteria will consume atmospheric CO2 and either store it or produce biofuel and foodstuffs. The Earth's temperature will fall back to normal level, and it also prevents ocean acidification, which increases the ability of marine life to extract calcium from the water to build their shell and skeletons. Secondly, microbial communities and methane cycle. Methane is a greenhouse gas most of the time enters to atmospheres because of microbial action. Methanotrophic bacteria is consume methane as their only source of energy and convert it to carbon dioxide during their digestive process. These bacteria can consume huge concentration of methane, which is helpful in reducing methane emissions from methane-producing factories and landfills. These include methyllocinus, methylocytes, methanomonas, methylomonas, methanobacter, and methylococcus. Next, in anaerobic conditions, carbon dioxide easily changes into methane that is accomplished by methanogenic bacteria. The conversion process needs hydrogens, yield waters, and energy for methanogens. To accomplish the recycling pattern, another group of methane bacteria called methanol oxidizing bacteria or methanotrophs can convert methane to carbon dioxide. In the presence of oxygen, CH4 is oxidized to CO2, yet use water and energy by methanotrophs. Methanotrophs tend to live at the boundary between aerobic and anaerobic environments. They have access to the methane produced by the anaerobic methanogenic bacteria, but also access to the oxygen needed for the conversion of the methane. Let's have a look at the overview. The bacteria that we mentioned just now consume CH4, thus reducing the methane emissions to the atmosphere. This is the fastest yet most effective way to slow the rate of warming now, as less heat trapped in the atmosphere, which slow down the melting of the iceberg. Lastly, we will talk about microbial communities and nitrogen cycle. Most of the time, microorganisms push the system in order to harvest energy or to accumulate nitrogen in a form needed for their growth and development. The major transformations of nitrogen are involved through the following steps. First, nitrogen fixation. Nitrogen is in the form of a very stable molecules which is unusable by plants and animals without fixations. Therefore, two kinds of nitrogen-fixing bacteria are recognized. The first kind, the free-living bacteria, includes cyanobacteria or blue-green algae, nostocs, azotobacter, and Clostridium. The second kind comprise the mutualities bacteria, mainly rhizobiums associated with leguminous plants. Nitrogen fixation is carried out by free-living and symbiotic microorganisms in a good manner. These bacteria have the nitrogenase enzyme that combines gaseous nitrogens with hydrogens to produce ammonia, which is converted by the bacteria into other organic compounds. Secondly, nitrification is the process ammonia transformed into nitrates by living things. Nitrates are what the organisms can absorb. The transformation of ammonia to nitrate is completed by soil-living bacteria and other nitrifying bacteria. In the primary stage of nitrification, the conversion of ammonia to nitrites is done by bacteria such as Nitrosomonas species. Other bacteria species such as Nitrobacter are responsible for the oxidation of the nitrites to nitrates. Third, assimilations. This stage indicates that the mechanisms of plants get nitrogen. Plants can obtain nitrates from soil by their root hairs. Eventually, it is used in cellular component production like amino acids, nucleic acids, and chlorophylls. In plants, they have a symbiotic relation with rhizobium. Some nitrogen is assimilated in the form of ammonium ions directly from the nodules. Other life forms also seek nitrogen through food chain structure. Ammonification is the stage of decaying. During living things are die, decomposers like fungi and bacteria turn nitrogen to ammonia. Later, it can re-enter in the normal nitrogen cycle. The process is termed as ammonification or mineralization. Many types of enzymes are involved, for example, GLN synthetase, GLU2 also glutarate aminotransferase, and GLU dehydrogenase. Denitrification. At the end of cycle, extra nitrogen molecule in the soil move off to the atmosphere. Denitrification is the reduction of nitrate back into the largely inert nitrogen gas for completing cycle. This kind of task is performed by special and unique group of bacteria like Pseudomonas and Clostridium. They use nitrate as an electron acceptor in the place of oxygen during respiration. The denitrifying bacteria use nitrate in the soil to carry out respiration and consequently produce nitrogen gas, which is inert and available to plants. The process takes place in the absence of oxygen commonly in waterlogged soils. Eventually, nitrates is converted to nitrogen gas and re-enters the atmosphere. Overview. The air and water can become less polluted due to less emission of nitrogen. Less nutrient pollution has impacted many streams, rivers, lakes, bays, and coastal waters. This will reduce environmental and human health issues and slowly revive the economies. Secondly, it also prevents algae blooms. This will restore the water quality, food resources, and habitats. It also increases the oxygen that fish and other aquatic life need to survive.